One of the striking things about the COVID-19 pandemic has been how suddenly it turned the world upside down. In early January, very few people even knew about the disease, and yet several months later, whole economies were being shut down. And during this time, decision makers have had to make choices about what to do in the pandemic based on limited information. There's still very many things that we don't know about COVID-19 and about the SARS-CoV-2 virus that causes it. For example, we don't know what the precise rate of spread in the population is. We don't know exactly what the case fatality rate is or exactly how the immune system responds to it. But there is one thing that we know with a relatively high degree of confidence about the SARS-CoV-2 virus. It very likely jumped into humans from another species recently and very likely spent much of its evolutionary history in bats. So how do we know that? Let's begin by considering a population, a hypothetical population where the coronavirus is spreading. In honor of Bill Purvis, who was the first biologist at Harvey Mudd and who passed away recently, we'll use these cute little guys. Purvis called them grudies and frequently made use of them in introductory genetics problems where their extreme fecundity came in useful. It turns out they're also susceptible to coronavirus. So imagine we have a sample, a set of infected grudies, and we take samples of the coronavirus from them. So one of the things that we could ask about those samples is how are they related to each other? In general, the samples will be related to each other based on the history of transmission that produced them. And in general, that's something that we don't know. But for the sake of kind of illustration, let's imagine that we're all knowing and that we somehow know what the whole history of transmission from one grudie to another of a virus was in this population. In that case, we would be able to, based on that knowledge, be able to draw a diagram representing the relationships of the different strains. This kind of diagram is in fact called a phylogenetic tree, and it's really like the classic diagram of biology. So it represents, in this case, representing the relationships between a set of strains. In fact, it can apply not just to viral strains, but to all kinds of organisms. And when Charles Darwin was first coming up with his theory of evolution, like in the first few months, he drew a picture of a phylogenetic tree in his notebooks. So the phylogenetic tree consists of nodes. An important part of them are the nodes. There are tip nodes or leaf nodes, as they're known as, and these represent particular samples or strains in this case. Then there are also internal nodes, and what internal nodes represent is their common ancestors. So for example, this internal node that I've labeled 3 is the common ancestor of B, C, and D, which they don't share with samples A and sample E. So we could call this the most recent common ancestor of B, C, and D. So here is another most recent common ancestor. This is the sort of internal node that is the ancestor to all the strains in the tree. This is also sometimes called the root. And we usually think of time as moving from the root toward the tips in the tree. And so that means that node 1 occurred before node 2, which occurred before node 3. And we can use that order of occurrence to help us understand the relatedness of different strains in the tree. For example, strain B and strain D share this particular most recent common ancestor. Strain A and strain B, on the other hand, their common ancestor is back here. And because the common ancestor of A and B occurred before the common ancestor of B and D, we can conclude that B and D are actually more closely related to each other than B is to A. So there are lots of ways of drawing trees. We can draw them in a square way, we can draw them in an angular way, we can have them pointing in any direction. So here's the tips are above, in this case right here. We can draw them radially, but it all amounts to the same thing. So we're not, all-knowing. We don't kind of usually know what the history of transmission in a population is. So typically we get some samples, and actually if we want to know the tree, we have to infer it from the samples. And for viruses like this, the way that we can do that is to compare their genomes. So this is a representation of the genome of the SARS-CoV-2 virus. SARS-CoV-2, again, is the virus that causes COVID-19. It is about 30,000 nucleotides long. So that is, it has sort of 30,000 nucleotide letters, A's, C's, G's, and so on in it. For comparison, the human genome is more than 3 billion nucleotides long, so the viral genome is quite short, and it consists of a number of genes that carry the instructions for making proteins. And so here I've got some example sequence that comes from one of them, sort of the S or spike gene. And so this is coding for a protein called the spike protein, which is actually on the surface of the viral particle. And it's involved in getting the viral particle into human cells. And it's also, importantly, very often the target of the immune system when people develop an immune response against the virus. So how can we reconstruct a tree? So very useful thing is that in that history of transmission, typically mutations are arising, and we can often make inferences based on that. So here again is sort of my scenario of transmission in this population of Grudies. And so during the course of this viral transmission from one Grudie to another, sometimes mutations arise. So here I'm imagining that there is a U to A mutation arising at this point. And what's going to happen is that the viruses which infect this Grudie and which get passed on from one to another will end up all inheriting that mutation, so that the samples that we look at the end, 4 of them are going to have that A mutation present, but the 5th will not, because this one down here is not descended from this point here. And again, if there was a mutation that arose here, for example, say a U to G mutation at this point, the samples that kind of descend from that virus are going to end up having that mutation, and other samples will not. And so this sort of pattern of mutations potentially makes it possible to infer things about the history of transmission. So, to be concrete, let's imagine that we have 4 samples from coronavirus that we've taken from different, say, patients, and we want to see if we can infer the tree based on that. So here what I've done is I've taken some sequences and I've lined them up so that kind of equivalent positions form a column. And I've also colored the sequences in a way where you can see where the differences are. So this representation is called an alignment, and it's a typical sort of starting point for making a phylogenetic tree. Very often the data that you're using to make a phylogenetic tree is an alignment like this. There are many ways to reconstruct phylogenetic trees. Many important examples are probabilistic, and the maximum likelihood approach is one example of this. And so in maximum likelihood, the basic idea is that we want to calculate the probability of the data given a model. And the model in this case is a tree, a particular tree with a particular set of relationships between, say in this example that I've got here, W and X are closely related, Z and Y are closely related. So that kind of set of relationships is one aspect of the model that the tree represents. And another aspect is the length of the branches, which in these kind of trees, branch length corresponds to an amount of genetic distance. And so the particular length of the branch is sort of saying how much genetic change we're expecting to have happened over that lineage. So given this model of evolution, we can calculate the probability of the data that we observe. And this is based on knowing something about the probability of one nucleotide replacing another, and about sort of the way sequence evolution works. It's possible to calculate this kind of a probability. And we call a probability like this, which is the probability of some data given a model, a likelihood. And a very common approach to reconstructing phylogenetic trees is to calculate likelihoods for different trees, and then we can explore the space of possible trees and use some optimization methods to try and find the tree which gives us the highest likelihood. And so in my example, here's that tree. And so this is sort of the maximum likelihood tree for this data down here. And so in this tree, X and Z are sort of most closely related to each other. And we also have got a set of branch lengths. So I've written the branch length, the number of the branch length next to the branch here. And this is in substitutions per site, which represents basically kind of the number of changes on the branch divided by the number of sites in the alignment, or the sort of length of the alignment here. So that tree that I just made, the kind of example that we just went through, I was imagining basically that we're using SARS-CoV-2 sequences. So samples that come from this the current COVID-19 pandemic. So here I've made a tree that includes one SARS-CoV-2 sequence, but also sequences from samples of virus that cause different diseases in humans. So this one right here, this is the SARS-CoV-1 virus. This is the original virus that caused the SARS pandemic in the early 2000s. There's something called MERS, which is related. This is the virus for that. And then here are a couple of viruses that actually cause colds. They're coronaviruses which cause colds and have been in the human population for some time. So I constructed a maximum likelihood tree based on those samples, and this is what it looks like. Now, one thing to mention as we're sort of thinking about building bigger trees that include more things is that actually coronaviruses fairly easily cross species boundaries, which I'm illustrating here in a kind of Grudy scenario where I've got two species, a blue and a purple Grudy, and I imagine that the virus passes, at this point, transfers from one species to another. We could call that a host transfer, going from blue to purple Grudy. And in fact, it seems that that kind of thing is pretty common in coronaviruses in general. So this is a tree based on real data, so taken from the literature, so that I've got the citation down at the bottom there. And this is just the kind of tree that we've been looking at. So the tips here represent samples of coronavirus that were taken from a particular individual or a particular animal. And the figure's also been labeled with the type of host, so human or bat, and so on. And so on this tree we have, for example, these viruses right here. These samples are the SARS-CoV-2 coronaviruses. Up here we have the SARS viruses. Down here is MERS, and here are those cold-causing coronaviruses that I mentioned. But notice that interspersed between these are a number of other viruses that are in fact infecting other things besides humans. And I'll come back and talk about sort of interpretation of that in a minute, but the first thing I'd like to consider is something about the timing in the tree. Something that might be interesting to know is, when did these various evolutionary events that are represented in this tree happen? So for example, right here, this is the most recent common ancestor of SARS and SARS-CoV-2. And it might be interesting to know, how long ago did that ancestral virus exist? And so let's say a little bit about how we can figure out dates in a tree. So here I have an example tree, and notice in my example tree that the level, the horizontal level that the tips come to is not the same for different tips. And there are a couple of reasons why this can be the case. One of them is that with pathogens, like the coronaviruses, typically the samples are collected at different times. And so in my case, I'm imagining the samples were collected over a 15-year period from 2005 to 2019. So let me define one term, which is the cumulative branch length for a tip. And so what we mean by the cumulative branch length is the sort of, it measures the total distance, the total branch length from that tip back to the root. And so I'm illustrating it here for tip D. The cumulative branch length will be the sum of all the branches that I've made red here. And so what does cumulative branch length measure? So remember, branch lengths represent an amount of genetic change. So the cumulative branch length is sort of telling us the total amount of genetic change from the root all the way to a particular tip. And there's we can calculate that for each of the tips potentially. And so here I have a graph where I've done that, where I've plotted out, in fact, cumulative branch length against collection date. And in general, we expect that there should be an association between these two things. The reason being that, if you collect a sample later, there's been more time for genetic change to accumulate. And so therefore you would expect that there should be more difference between whatever sample arises then and the root. And so in general, we expect there to be an association, and in this example there is. You might ask, why do they not fall in a perfect line? And the reason for that is that the sequence change is a random process, and in fact it's possible for the rate of that process to be different in different lineages, which will sort of introduce noise. Okay, so with this plot of cumulative branch length versus collection date, one thing we can do is we could fit a line to it, and if we follow that trend line to the point where it crosses the x-axis, which is the point of 0 cumulative branch length, that's going to give us an estimate of when the root should have existed, right? And so in this particular example, we would conclude that the common ancestor of all these strains likely existed between 1970 and 1980. So this method that I've just described kind of gives you a flavor of how dating methods can work. In many cases, when people are doing research these days on these kind of questions, they're using some more sophisticated probabilistic methods. And so the data that I'm about to show you, that's the case for the data that I'm about to show you. So this is a tree that comes from a recent paper. It's sort of my distillation of it. I've taken the tree and simplified it a little by removing some strains so it's easier to look at. But the basic emphasis of this study was where one of the main things they wanted to do was look at the dates in the tree. And so here we have SARS-CoV-2, right? So the virus that's causing the current pandemic, SARS is up here. And then we have a number of other strains that infect things including bats. So this closest relative of the SARS-CoV-2 virus is another virus which happens to infect bats. And the estimate for the best estimate for the date of divergence between these two lineages is that it happened in the late 1900s. If we step back a few branches in the tree, we get to a point where there's a branch going off that leads to a virus that infects pangolins. And that common ancestor is estimated to have existed sometime in the mid to late 1800s. And if we step back still farther to this point, this is the common ancestor of SARS-CoV and SARS-CoV-2, which was the question that I asked a little while ago. In this study, the estimate that they came up with for when this common ancestor existed was around 1200 AD with an uncertainty of some hundreds of years on either side. And so the takeaway message from this tree is that the coronaviruses are, relatively recent. So dates on this tree are all quite recent. And so that's, interesting because it shows you that the rate of evolution of coronaviruses, and this is true of many viruses, is quite rapid compared to the rate of evolution of their hosts. So in the case of the coronaviruses, they're infecting mammalian hosts, things like humans and bats and pangolins. And those mammalian hosts, don't change nearly as quickly as the viruses do. So in 1200 AD, humans were still humans, bats were still bats. But actually, these coronaviruses were quite different at that time. And so one question, that we might ask is, well, what host was the virus infecting at that time, back when this common ancestor existed? And so to illustrate how we can reason about that, let me go back to a Groody example. So here's a very simple tree for coronaviruses that are infecting Groodies. And on the tips, I'm showing what Groody is getting infected by the particular sample. So we have a couple that are infecting the blue Groody and one that's infecting the purple Groody. And so a virus, at any given time, a virus could be infecting one particular host. And when we think about a lineage of viruses, they're infecting a particular host, but that lineage could potentially jump to infect a different host. It could potentially change. And so in reasoning about what's happened in these lineages and in this tree, we use one kind of important principle, which is parsimony, to try and sort that out. And parsimony is the idea that scientists use to choose between different possible explanations, and the idea basically being that in any explanation that we come up with, we should try to appeal to unlikely events as little as possible. We want to minimize the number of unlikely events. And so with this kind of data, this tree that I'm showing you, one thing that we could say right off the bat is there are two different Groody species here. So it's going to be parsimonious to assume that the ancestor was in one of those two. So that sort of gives us a kind of a starting point, and we can come up with a couple of scenarios. One scenario is that the ancestor infected the blue Groody at the root. And so that's my scenario one here. And in scenario one, we're imagining that, well, it's blue at the root, and it just stays in the blue Groody all along this lineage. And when we get to the tip, it's still in the blue Groody, which is good because that's what we observe in the data. And similarly, along this lineage, it can just stay in the blue Groody. But on this lineage up here, the tip is actually in the purple Groody, so we're going to require a host transfer event from blue to purple. right here. So in scenario 1, we have one host transfer event. The other scenario, of course, is that we started in the purple GRUDY, and in that case, we're going to have a couple of host transfer events. We're going to have one host transfer event here, where we go from purple to blue, and we're going to have another one along this lineage, going from purple to blue as well. And then the third branch here can just stay purple. So for scenario 2, we're going to have 2 events, and that means that between those 2 things, we would favor scenario 1 because it requires less host transfer events. So we have host transfer events we think of as being relatively unlikely sorts of events. We'd like to have a good explanation should have less of them, and so scenario 1 is preferable because it minimizes the number of host transfer events. So here is this tree that I've distilled for you again, and we have humans, bats, pangolins on the tips, and we're kind of wondering about, for these internal nodes, what do we think was the host that the virus was infecting at that point. And so this tree's got a lot of bats on it, and in fact, when you go through and think about so what parsimony would suggest, it suggests that the common ancestors in this tree were infecting bats in most cases. And if we look at the sort of stuff that's closer to SARS-CoV-2, these few divergences, like the most closely related things to SARS-CoV-2, are also in bats. And so that suggests that in its most recent evolution, the virus was probably evolving in bats. Now there's some possibility that in the kind of very, very short term kind of transfer to humans may have involved some intermediate species. But based on this tree, we would conclude that kind of on the scale of 10, 20, 30 years, it's very, the virus was mostly evolving in bats during that time. We'd also conclude from the tree that the common ancestor of SARS-CoV and SARS-CoV-2 was also likely in bats. Now, there are sometimes some ambiguities that the parsimony approach leaves us with. For example, in this tree, I've colored it so that I'm sort of imagining that it was bats all along here. In fact, it would be equally parsimonious to imagine a transfer event into pangolins at this point and then back to bats over here. But that notwithstanding, it's clear from the tree that coronaviruses are basically bat viruses. And in fact, people are these days very interested in trying to find additional coronaviruses in bats to see what we can learn. Researchers are thinking that there's probably a decent amount of viral diversity that exists in bats that we don't know about yet, that we haven't sampled. And given the fact that we've had three sort of deadly outbreaks of coronaviruses in the last 20 years, it seems like exploring that diversity and kind of getting a better sense of what's out there would be a good idea. So I'll point out that this kind of reasoning works for other pathogens besides coronaviruses. So here's an example. This is a tree of HIV-1 sequences. And so on the tips, these viral sequences, they were taken from humans, chimpanzees, and gorillas, so 3 different primates. And then I've colored, sort of based on the parsimony approach, I've colored the different branches, as to what we would infer these ancestral viruses were infecting. Green is chimpanzee, orange is human, and gray is gorilla. And so from the parsimony-based logic, we could conclude that HIV-1 is, ancestrally was in chimpanzees, and that it entered the human population independently a number of times. So, and that's interesting because that means that some strains of HIV-1 are actually more closely related to chimpanzee viruses than they are to other strains of HIV-1. We can also use this same logic to make inferences about sort of the more recent spread of the virus and what countries it's been in. So instead of mapping a host onto the phylogenetic tree, we could actually map the country where the sample was taken. So here I have 7 samples of SARS-CoV-2. So this is just the COVID-19 pandemic viruses. And we're indicating what countries these samples were taken in. So we have some from China, some from Europe, some from the US. And so then I've gone through and I've colored the branches according to which country we would infer that the ancestral virus was in. So I'm making purple correspond to China, green is Europe, and red is the US. So one takeaway from this tree, kind of immediate takeaway, is that because many of these early branches are Chinese, this is entirely consistent with the idea that the known fact that the pandemic arose in China. So this is exactly what you would expect from an infectious disease that began in China. We can also learn some interesting things about the viruses which are in the U.S. and how they got here. So there are two different samples, of course, that were taken in the U.S. on this tree, and they actually fall in different parts of the tree. So this sample down here came from Washington early on in the epidemic there, and you can see from the tree that actually the evolutionary history of this particular sample suggests that it likely came from China. So that the lineage that leads to this sample probably came directly from China because it nests in with the Chinese sequences. In contrast, so this is on the west coast of the US. In contrast, here's a sample that was collected in New Jersey on the east coast, and this particular sample actually nests with the several European samples. And so this is sort of showing us that many of the East Coast samples and many of the viruses that are causing the outbreaks on the East Coast actually had a European origin, and at some point in their evolutionary history had passed through Europe. So here's a more kind of detailed tree showing the same thing. So this comes from a website called nextstrain.org, which you can actually go to that website and build trees for yourself if you like. The coloration is the same as what I was showing on the previous slide, but it just includes way, way more samples. And so to return to our original question, the way that we know about the history of SARS-CoV-2 is through phylogenetic trees. We can build those trees by comparing genome sequences from different strains, and then we can map onto the trees information about viral host or about country of origin, and on that basis make inferences about the history of the virus. And I'll just close by noting that in some quarters people have suggested that this outbreak might somehow be due to human manipulation. And I think that the methods that we've been talking about and the data that we've looked at show that this is unlikely. SARS-CoV-2 could be explained very well on the basis of natural events. It's likely that the virus evolved in bats and then transferred into humans via a host transfer event of the kind that's common in coronaviruses.